Okay, welcome to part two. Let me confirm I started. Yes, I did. Okay, part two. We're gonna get a little more detailed on the parts of the plants now. We're gonna get into different types of plants. First thing we're gonna do is we're gonna start out with on page 436. Again, if you don't have your book with you, pause the video, run and get your book, grab it because you're gonna use it. There you go. I gave you time to get your book. Okay, so there's a cross section of a leaf there with the cells. If we were at school, we'd be looking under the microscope at this right now. But it's a good picture. So, a couple of things to note on here. The cells, the plant or the leaf has an outer layer of cells on the top and an outer layer of cells on the bottom with something a little bit different that we're going to talk about here. If I can draw it halfway decent, we'll throw one right in there. Okay, these are called the epidermis. Just like your epidermis, it's the skin. The epidermis tends to be it's got more of a waxy coating on it to keep water actually in this plant, not necessarily out of it. Leaves are so thin, they worry about losing too much water. Inside the epidermis is top and bottom. Okay, are two types of cells called mesophil or mesophil, depending on who you talk to. First type is called the colonnade mesophil or mesophil. It forms columns, long columns. It's a long drawing. It's like he's got a ton of teeth. Okay, so palisade, not colonnade. Palisade mesophil. They look like columns. This is where most of your photosynthesis occurs. They're long so that the leaf points towards the light, the sun. The top of the leaf, when it can move, it's got a little thing called what is it called? I want to say phototropism, but that's not right. But it can move to face the sun, and so they're longer. So as the sun passes through, the leaves are very thin. It has lots of chlorophyll that can catch that. Underneath are what's called the spongy mesophyll. Why this is spongy is it's got cavities in it. The cells are all not right up next to each other. Some are close, some aren't. There's a reason for this, almost as if it were designed. What's going on here? We got our spongy mesophyll. What works in conjunction with is along the bottom of the leaf are what are called stoma, or holes, pores. And these stoma are guarded by little cells called guard cells. These guard cells open and close the stoma so that it can allow air in and out or not. What this does is this allows for evaporation, or what's evaporation in plants called? Called transpiration. Transpiration is very important for its circulatory system, basically for its phloem and its xylem. Because a tree does not have a pump, it doesn't have a heart that pumps it through. So what happens is transpiration happens and water exits out the leaves. When that water exits out the leaves, it creates a pressure that pulls water from the veins in, and that comes up the xylem. So through the xylem works its way down the stem into the roots, and it pulls water out of the ground. So it kind of creates a pressure that allows water to flow up the plant. Phloem, going the other way, just uses gravity primarily to move things around. So, okay, so spongy mesophyll, palisade mesophyll, all these cells can photosynthesize. So they all have that capability. These are designed for the highest rate of photosynthesis. got a vein in there, so with xylem and phloem, or with xylem and phloem, but not in here. So that's a cross section of a leaf. Know those parts. Know your epidermis, your palisade, how to tell the difference between your palisade and your mesophyll, and know about stoma and Gart cells and why it's necessary. Okay, so let's move on. Okay, so the main reason for the leaves is photosynthesis. You should be kind of familiar with photosynthesis by now. We've talked about it before in other classes. Photosynthesis is synthesizing sugars from sunlight, carbon dioxide, and water. So the plant builds its sugars. What's nice enough is it's nice to produce a lot more food than it'll ever use so that we can pass it on to other creatures and eventually to us. So, okay, so the main thing that carries out photosynthesis is something called a chloroplast. It's one of the plastids. Plastids are basically organelles that contain pigments. Pigments contained in there are generally called, I can't even say this, carotenoid. I can't even spell it. I used to be able to say that. Carotenoids. Basically, carotenoids are usually orange and yellow. Chlorophyll, the main one used in whatever we're talking about, in photosynthesis, is green. So what happens is, there's other ones called anthocyanins. You should be familiar with anthocyanins, or you will be very shortly when you take chemistry, because we use an anthocyanin found in red cabbage as a pH indicator. So what happens, there's a pH, determine the pH color of, or pH to help determine leaf color. And in here they use red cabbage, and they change the color. And the anthocyanin and red cabbage, what will happen, first of all, pH, you're probably familiar with, actually it's capital H. pH, you should be relatively familiar with by now, is acidity or basicity. Neutral is 7. Acids are less than 7. Bases are greater than 7. Now what's cool about the anthocyanin and red cabbage is acids. It starts out, it's blue. Blue is neutral. If it becomes an acid, it turns pink. If it becomes basic, it turns green and then yellow. So it's kind of a fun indicator. We'll play with it if we don't get a chance to play with it in biology. For sure, next year if you take chemistry, we'll play with that. So this is the reason that leaves change color in the fall, because they start dying off as the pH changes inside of the leaf. Things like chlorophyll break down, you're left with your oranges and your reds. Anthocyanins or things like that can react with the chemistry of the leaf itself and change its color as well. So okay, that's enough about that. Leaves, you'll read a little bit more about leaves. You'll get to deciduous plants that lose their leaves in winter. We're very familiar with that up here in the north. So trees, what happens is, remember our leaf? We got our petiole and we got our stem. In between the petiole and stem is a layer called the abscission layer. I want to make sure I'm spelling it right. Abscission layer. This layer, when the time gets right, it gets the right chemicals, it'll say, okay, time to break down. So that abscission layer breaks down, the leaf falls off. It kind of shuts down. It does other things as well. It's cutting off sap, things like that. So You'll read a little bit about the abscission layer. Okay, so that's enough about leaves. You know pretty much about leaves. The other part we want to talk about is roots. What do roots do? They absorb water, absorb nutrients. Can be used for food storage. So when photosynthesis goes about, remember it takes carbon dioxide and water with energy from the sun to give us glucose and oxygen. So that's not balanced, but don't worry about it, that'll be chemistry. Okay, so glucose. You all know glucose is a very sweet sugar. Used in some pops now, more natural. We got fructose and sucrose, which are bisaccharides. But when the plant wants to store this, it'll take a bunch of them together. So each G represents a glucose. It'll start banding them together. The longer this chain gets, the less sweet it gets. And it goes from being a sugar to a starch. So it stores that food for making it through the winter, and if you think about it right, it's a design so that that energy can be passed on to other creatures, animals, things like that can't produce their own food and need a food source. So that starch is stored. One of the places it likes to store that starch is in the roots. You're familiar, potatoes, carrots, beets, all kinds of things. Pineapple. I think pineapples. I don't know if it's a root or not. That one could be a little tricky. But so it stores it in the roots. Now you got leaf identification. There are some root identification. Does it get into monocots and dicots yet? Let's see. I didn't look. I don't think so. I think it gets into monocots and dicots next week. How plant depends on water absorption. Huh. Let me see. Sorry for the little thing. Large woody stem. No, it gets going in monocots and dicots. So, two types of roots. Taproot. It can still have some extra roots coming off. And fibrous root. Food storage happens in taproots, because that's where it does it. Otherwise, there's things called tubers and things like that. A potato is actually a fibrous root with tubers on it, which are storage points. So, taproot and fibrous. Easy to tell them apart. Now, the reason they're going into this identification is because plants are can be classified as two types. You got a monocot and a dicot. Now it gets its name from what's called a cotyledon. On a seed is an area where food is stored called the cotyledon. You got the embryo and you got the food source. A monocot would be corn. If you look at a corn seed, it looks like this. It's got one, this is a cotyledon. It's got one cotyledon, mono, single. Dicot, a pea plant. If you ever looked at a pea plant, especially look at it from the side, you'll notice that there's a line down the middle, then you got your embryo down here. These both are cotyledons. That's a dicot, di2. So monocot, one cotyledon, dicot, two cotyledons. Turns out there's some other things that go along with it as well. You got your book handy again. Let's see. Let's turn to page 445. Let's go 444 real quick. Just real quick, this looks at a onion root tip, one of those places of meristematic growth. If you look on there, it's got a meristematic region just above the root cap. The root cap is basically just a bunch of cells that are there to push through the dirt and possibly get damaged and torn apart. So under that is the meristematic region. That's where all the growth is really happening fast. So And then you got the elongation region where those cells start growing, get longer, and then the maturation region where the cells are about what they're going to be. So just quick there, we would look at that under the microscope again if we were there, to point out and look at that. We've done it before if you've looked at different stages of mitosis. That's a good place to look at it. On the next page, page 445. Says a lateral cross-section of the maturation region of a dicot root. One of the difference between monocots and dicots is the vascular tissue. In a dicot, if you look at that vascular tissue, they zoom in, and there's an X. They will form an X in their vascular tissue. That is a xylem. The phloem is around the outside, away from the X. So that's a dicot's vascular tissue. Turn to the next page. It says magnified cross-section of an herbaceous monocot system. So if you look at that, some people say they look like little monkey faces. I'm not sure. But the vascular system in the dicot is in the center. The vascular tissue in the monocot is scattered throughout the stem. And you see little vascular bundles there. I believe you'll get a picture of that either in your study guide or on your test. So I'll be able to identify a monocot vascular bundle. Okay. Okay, so that's another difference. They're talking about root stems, herbaceous versus woody. I think you can figure that out. Woody, tree, hard, tough. Herbaceous, easily squished or cut. So let's see. On page 447, you can look at the different parts of the woody stem. You got the meristematic region on the outside. Inside, you have the pith, or the kind of just the cells that hold everything together, and the vascular is near the outside as well. That's why it's hard on a tree if it gets girdled. So if you have a tree, the vascular system runs around the outside. And if you get something tighter on there, it can cut off that vascular system and kill the tree. Sometimes the roots can actually do that. They can kind of grow around the tree. And as they get bigger, they can cut off that vascular tissue and kill the tree. So. Okay, so cross section of roots and stems, that's fine. Okay, kind of running out of time. You'll get into classification of plants in bryophytes, seedless plants, seed-making plants, on to the end. If you have any questions, there are more things for the monocot and dicot. I don't know if they get into it. Venation or leaf venation is parallel. Well, here tends to be palmate here. There are other things. Go ahead and make a list if you want of the differences between dicots and monocots. If you have any questions on the differences, I believe there are 5 differences if I remember right. If you have any question on the difference, drop me an email and I'll tell you the differences on that. But it'd be good if you can kind of gather that together. I know that's on there and they don't just come out and just put it all in one place for you, which would be nice, but then you know the material. So. Again, if you have any questions, drop me an email, Mr. C at mrcscience.com, and if there are enough people with questions, we'll get together a Zoom meeting and talk about it. Talk to you later.